All right, ladies and gentlemen, here's a little impromptu instructional video for y'all. Because I wasn't planning on being out, but my babysitter, I guess you could say, canceled on me on the last minute. My wife is doing some all-day training stuff that she has to be a part of. So I will be working remotely for the day, going through stuff. So I put this together real quick for y'all to go through some of the neuron stuff that I was lecturing in person during A-Day. So you'll want to take some time to watch this information. And then I talk about the modeling activity that I have linked in the agenda after this one. So have some notes handy. At the very least, you need to have your intro to the nervous system notes. Then you'll add to some of this stuff. If you want to access this slideshow, you can click the link in the agenda to pull this up, make a copy of it, or download it so you can incorporate this into your notes also. So let's go ahead and get started and talk a little bit more about neurons. Okay, so we're talking about neurons, and specifically we're going to be looking at this thing called action potentials. Now, we mentioned that a lot when we were talking about muscles a couple weeks ago. And so what we want to do is we want to focus a little bit up on how neurons send information to different parts of your body. Okay, what do we mean when we talk about sending nerve impulses? Okay, so to get us into this, the nervous system is made up of two divisions. Okay, the central nervous system, which is the brain and the spinal cord back here, and the peripheral nervous system, which is all the other nerves that branch off of the brain and the spinal cord. Okay, there's other divisions that I mentioned in the first lecture video, and I'll bring those up again through this one. Okay, so just kind of have those ready to go. So this is the basic structure of a neuron. And there's three basic parts. Now, you know from the notes that this isn't the only nerve cell structure that you can have. This is by far the most common one. And so this is the one that we're going to stick with. So it comes in three parts. Okay, we've got the cell body, which is this part right here. We've got the dendrites, which are these parts over here that look like trees with the leaves that have fallen off, excuse me. And then we have the axon, which is this long section right here with this other tree-like portion that's called the axon terminal. This particular neuron is a motor neuron that we had talked about when we talked about neurons with muscles. The dendrites are the receivers of the message, and the axons send the messages. Then the cell body has all of the nucleus and stuff like that. Okay, so most of these neurons that we're going to be talking about are myelinated. And myelin is this sort of wax-like substance that wraps around the axon. Now, the axon is a very long extension that sends the impulses. That's still part of that one cell. Your sciatic nerve is the longest nerve in your body. And the diameter, I think we determined earlier on Monday, that it's 3/4 of an inch thick, so it's pretty sizable. And so this fatty or wax-like substance is known as myelin. And there's specific neuroglial cells that make these, and those are known as Schwann cells. And so each little section of myelin, if you think back to the picture from the previous slide, they look kind of like little rolls of insulation around the cell. And the main part of the cell is right there. That's where the axon is, and then all those other layers around that is the myelin sheath. You can take neurons, you can classify them into three different types. Okay, you've got sensory neurons. Those are going to be your neurons that carry sensory information and nerve impulses and stuff like that to the brain. So like here's touch receptors in the skin. Those are sensory neurons. They're going to take information to the brain. Once in the brain, the information is going to be carried by relay neurons, also known as interneurons. So here's your interneuron. Those are known as your relay neurons. Those are the ones that are inside your brain. They're sending information from nerve cell to nerve cell, brain cell to brain cell, as you're processing information. Then once that information is going to leave the brain, it's going to go on, most commonly it's going to do motor neurons. Those are the ones that are attached to your muscles right here, forming that neural muscular junction that we talked about when we were looking at muscle contractions. Okay. You can have, so this also, they're similar to this, to that particular process going to your brain, is the reflex arc. Now, your sensory neurons are going to be part of the afferent division. So bringing information to the brain, where it's going to be integrated in the brain. Now, in some true reflexes, you can actually skip the brain and go straight to the spinal cord right here. If you flinch, if something's coming towards you, if you touch something hot and you jerk your hand away without thinking, that's because sensory information went to your spinal cord, and your spinal cord spit information back to your muscle cells. The information going back to your muscle cells is going on the motor neurons or the efferent division. And then typically it'll go to the muscles, and we'll talk about this when we do feedbacks and stuff like that, which is your effector, what's affecting the change. Okay, so we're going to talk about this potential a lot. We're going to use this word a lot. And so what we're talking about is a difference in electrical charges between the outside and the inside of the membrane of the neuron. Okay. Inside the neuron, it's negative. Outside the neuron, it's positive. And so we end up with a resting potential. We're going to get a voltage because of those differences, okay? And so we call it potential because it's the potential to have some sort of activity going on, okay? And so we regulate this potential using various channel proteins, okay? And so one of the things that we do is involving active transport. And so with the ions that we're focusing on the most are sodium and potassium. Now, there are other ions involved, like chloride ions, other anions, negatively charged ions, but they're not the ones that are moving. And you'll see that when you do the modeling activity in a little bit. And so ATP is going to be used on a particular type of protein. It's going to move in 2 molecules of potassium, and it's going to move out 3 molecules of sodium. And so they get moved through something called the sodium-potassium pump. And so you need to think of it in terms of like a water pump or a fuel pump, okay? Something that uses energy to push a fluid, okay? To that help, an air pump, same way. Some sort of machine that uses energy to move substances, okay? If you can get that idea in your head, that's basically what we have going on right here, is we are pumping out these ions, and it's using energy. And so it's just a particular type of protein. You'll see some later on if you look for pictures of what these look like. Basically, you'll have something that kind of looks like this. Kind of almost like pliers or something like that. And they've got spots for the sodium. That's right there. They've got spots for the potassium. That's right there. Okay. And so what this is going to do is this is going to set up a concentration gradient. Okay, now remember way back in the day we talked about active transport and passive transport. Active transport moves substances against the concentration gradient. That means it's going to take a high concentration of things and it's going to force, in this case, sodium and potassium to a higher concentration. So it's going to increase the concentration. Nature doesn't want to do that. It wants to go from high concentration to low concentration. That's where the diffusion comes in. We've got facilitated diffusion. Diffusion is going to be happening through channel proteins, okay, that are going to allow sodium to come into the cell and pass potassium to leave the cell. That's what we have going on right here and right here. This thing right here is our sodium-potassium pump. Like this. So that's going to push things against the concentration gradient. And so what that's going to do is that's going to set up so we've got more sodium. Let me get rid of this. That's going to give us more sodium outside of the cell and more potassium inside of the cell. So then what's going to happen is these channel proteins are going to allow the sodium with the potassium to flow down the concentration gradient, go from high concentration to low concentration, okay? And so what this gives us is a net positive charge outside, like we mentioned earlier, and a net negative charge. Now, one of the words that you need to jot down, because we're going to use this a lot, is it is polarized, okay? We've got a positive end and a negative end. That's what that means. So our membrane is naturally polarized. Okay, that's the resting potential. If your nerves are not being activated, they're going to sit with a positive outside and a negative inside. Okay? Now, here's the basics of a nerve impulse. All right, that's the signal being sent down your nerves. Something is going to trigger the neuron. It's going to stimulate it. A touch, temperature, blood pressure, light, sound, smell, taste, whatever. Okay, that's going to open the sodium channels. That's going to allow sodium to come in down that concentration gradient. Okay, the membrane is going to depolarize. So remember, we had a charge on either side. We had a positive on the outside and a negative on the inside. Basically what that means is right now our membrane is sitting at about negative 72 millivolts. As it depolarizes, this number right here is going to become more positive. Okay, it's going to get closer to zero and then it's going to get greater than zero. Okay, while this one section, so we've got this one section right here, okay, of nerve cell, it's depolarizing. That's going to cause this section to start to depolarize, because sodium is going to start to come into these channel proteins. Okay, because remember, your axon is a long string-like structure, okay, like a cable or a fiber or something like that, okay? And so what we do is we chop it down like this so we can see this cross section. See this ring right here, okay, is our membrane where our channel proteins are embedded. Okay, I tried to focus in, that's about the best I can get. And so it's all happening right here. And so if we were to cut this up like you're dicing up a carrot, we would have these channel proteins. So we've got a section here, it's depolarizing. It's going to cause this section to depolarize. So sodium is coming in. We're going to have a section right here, sodium is coming in. Okay? That's going to cause this wave of sodium, 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 sodium. And then eventually potassium is going to change. So potassium, 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 potassium. It's going to move down our axon like a wave. Now it's going to look like the wave, okay, follow me on this one, and let me get there we go. So our wave, right, as it depolarizes, it's going to get more positive, it's going to reach a maximum, right here. Then it's going to go down like this, a little bit lower, and then it's going to come up like this, right? So here's our wave. And that may look familiar to you, especially if you've been in the hospital or you know somebody who's been in the hospital or you've watched doctor shows, okay? Here's a graphic showing the same exact thing. So the first part, here's our polarized membrane at rest. Sodium ions are going to be coming in. That's going to depolarize, okay? That's going to cause a wave-like motion moving down the neuron, okay? This is going to lead us into an action potential. The permeability of the membrane to sodium is going to change. Sodium is going to be able to pass through the membrane. Those channel proteins are going to open up. That's going to drive more potassium to start moving through as well. And so, and here it is again. We've got our sodium ions diffuse down a concentration gradient. The entry of the sodium ion disturbs the resting potential and causes the inside of the cell to become more positive. Here's that graph that I just drew. So right here, sodium ions are coming in, they're coming in, they're coming in, they're coming in, they're coming in. Sodium ion concentration is equalized. Potassium ions are going to come in, potassium's coming in, potassium's coming in, potassium's coming in. They're going to stop and the potassiums are going to equalize. And then this period right here, we're going to reset. Okay? No, just finished that one. All right, so as the cell is depolarizing, there's a threshold that you have to meet. A certain limit, okay, that the voltage, that number has to change to right here, okay? And if you stay if you get above that number, you will generate an impulse right here. If you're below that level, nothing happens. And I'll talk about that in just a second. Okay, these things happen really, really fast. And one of the ways that these happen faster, like in that sciatic nerve, that long motor neuron, is those myelin sheaths. So here's why those myelin sheaths are important, okay? These myelin sheaths have spaces where the axon is exposed. These are known as the nodes of Ranvier. So here, these little sections right here are the nodes of Ranvier. Okay, so in between the myelin sheaths. The action potentials will jump from one node to the next, like they're leapfrogging. What helps me think about it is you're running down the stairs, okay? What's faster? To go down each step one at a time, or to skip steps? Skip one, skip two, skip three, jump all the way down to the next landing, and then jump all the way down. Which one's faster? I'll give you a hint. It's the skipping. If you go downstairs skipping every other step, you can go down faster. Okay, and that's basically what's happening here. As you're skipping to these different nodes, you go faster. And this is known as saltatory conduction. It's skipping from one node of Ranvier in between the myelin sheaths to the next one. Now there are some unmyelinated neurons. Most of those are going to be in your brain. They are not going to go as fast as a myelinated neuron, but they don't necessarily need to go that fast because they're covering a really short distance. That's one thing. Okay. Other things that you can do to speed up the nerve impulses, the bigger the diameter of the axon, the faster it goes. The warmer the temperature, the faster it goes. If it gets too hot, then the proteins denature and everything falls apart. Now, back to that threshold potential thing that I told you about. Okay, so sodium is coming into the cell. that is changing our resting potential. It's making it more positive. If it does not meet that threshold, I want to say that threshold is in the neighborhood of negative 32 millivolts. Nothing happens. The stimulus was not strong enough to cross the threshold. We do not have an action potential. Basically, the body's saying, like, this isn't important. And so you don't notice it. If you do cross the thresholds, then an action potential happens, but it goes all the way. It doesn't matter if it's somebody has just poked you with a pin or a bug has landed on your arm. The action potential is going to happen the same way. Another way, a way to think about this one would be if you're listening to your headphones, but you've only got 2 volume settings, muted and max. That's it. Either you hear nothing or you hear it all as loud as it possibly can. That's how action potentials work. Now, if you haven't done the synapse thing yet, you should go through and do that, because I'll pause here for just a second. Synapses are kind of like gatekeepers. Okay, so let's say a stimulus is strong enough to trigger that action potential. So when you're sending that signal down that neuron, it's going to reach the synapse. Okay. If the action potential is not strong enough, or there are not enough action potentials, that's not going to, then you won't trigger the release of the neurotransmitter, which means you're not going to continue the action potential on the next adjacent cell, on the postsynaptic cell. And so it stops. Once again, you're not going to notice it. The body's going to say, like, no, that's not important. We don't need to worry about that. When we come back in Thanksgiving, we'll look at upregulation and downregulation and stuff like that, and how you can amplify or dampen neurological activity using the different neurotransmitters, but we'll get into that stuff later. Towards the end, so when we were looking at our so here's our resting potential right here, these flat lines, right? Depolarization happens at the top lines right here. The refractory period is a rest period that happens right here, okay? And so basically what's happening is the membrane has to reset itself. And so those sodium potassium pumps, the sodium potassium pump has to reestablish those concentration gradients. And so it takes a little bit of time to pump the sodium back out of the cell and the potassium back into the cell to reestablish those concentration gradients. So when we're ready to stimulate the nerve cell again, those channel proteins can open, allowing the sodium to come in on its own, following that concentration gradient, the potassium to leave, doing the same thing. And then we get back to the refractory period. We have to reset everything. Okay, so this is kind of like you set up dominoes. You set up all the dominoes are up in a row and everything like that. You push one domino down, the whole thing goes down in sequence, right? That's how action potentials work. I can poke myself in the arm right here, and the stimulus that is here goes to my brain, and then, well, in this case, nothing happens. It doesn't come back. But if I'm going to do something about it, ouch, flick, then it went to my brain and then it came back. Okay. What's nice about these action potentials is they're self-propagating. Just like you don't have to knock each individual domino down. You just start one and then the rest follow. Right? So once an action potential starts right here, it continues up my arm, up my neck, into my brain, where I process it as I'm poking myself in the arm and this hurts. Okay, I don't have to stimulate my nerve as I'm going up to know that I'm getting touched right here, which makes things super duper efficient. And other organisms they're decentralized like that. They don't have, they don't have that thing going for them. Okay. So now let me go ahead and I'll give you a second while I'm transitioning over. I need you to pull up the modeling action potential activity. It's linked in the agenda. You can also find it in Echo. Okay. So let's meet back here in just a second so we can pull those up. Okay, so here's the modeling activity that I was telling you about that you wanted to look at. So you're going to need a whiteboard, dry erase marker, and an eraser. You're also going to need one of the 4 different types of beans. So I've got beans up at the front of the room. There's enough for each group. You're going to need 4 bags, each one with a different type of bean in there. You're going to use those to represent the ions that are going to be involved in this process. Okay, so you're going to follow the steps as you're going through. Okay, draw a circle to represent a neuron. Draw the sodium-potassium pump right over here. Indicate what the beans are that you're using. You've got pinto, black bean, lentil, and garbanzo. Okay, just make sure that you've got dark color to dark color and light color to light color. Okay, set up your ions, like here, number 5. There's some guiding questions that you should answer as you're going through. I did not say, and you should do this if you haven't done so already, you need to make a copy of this so you can edit it. Okay. Then you're going to model the actual steps that it goes through. You're going to model the resting potential and then the action potential as you're going through right here, moving the beans around and stuff like that, and seeing what the numbers are. Because here's something that's really important. Okay, when we're looking at this, we've got chemical equilibrium, and we've got electrical equilibrium. Okay, and so electrically, our nerve cells are stable. Okay, chemically, they're not. Okay, and when we talked about diffusion at the beginning of the school year, we were talking about chemical gradients and chemical concentrations. Okay, and so your cell aims to be electrically working with electrical gradients. Okay, but it achieves that by working against what nature wants as far as the chemical gradients. Okay, and so the activity with the beans helps us conceptualize what's happening, because we're physically moving these charged particles with and against concentration gradients to set up electrical gradients and electrical equilibrium. Okay? What you're going to do after you've had a chance to model this, you're going to go through and you're going to put everything, all the steps that you're going through all together. And you're going to film a video clip explaining the action potential process using the manipulatives that you have available. Okay, the beans and the whiteboards and stuff like that. You're going to submit 1 file per group, and I put a link to the group assignments in the agenda. They're posted in Echo. Okay, so click that link so you can open up who your group members are. Each person has to have a part in the explanation process. Now, I'm sure you're already thinking there's some logistical things that you'll need to work through. Okay, one suggestion could be you do the filming or the picture taking or whatever you end up using. And assign parts. So if you've got asynchronous group members, assign that part. Asynchronous group members, I would recommend you previewing this, communicating with your group members, and start working out which parts you're going to say. What I'm looking for is does in your part, do you understand what's going on? And as a group collectively, do you understand the big picture about what is going on? Okay. I'm also going to be looking for clear evidence down here at the bottom that each group member participated. So there's a lot of different learning outcomes I'm looking at. Collaboration, oral communication, knowledge and thinking. And so in Echo, I ask you to tell me, along with the link, who's doing which part, so I can listen for those parts. Okay. And that's it. This is going to be due for y'all, to give y'all the same amount of time. It's going to be due on Friday. I would recommend trying to get it done. Use your time in class to get as much stuff done as you can as possible. So that way all you have to do is just stitch everything together and send it to me, let's say before Thursday. But it's not technically going to be due until Friday at 11:59. Okay. If you have any questions, let me know. I will be monitoring messages, so you can send me a remind message if you need to. If you've got questions and stuff like that. I won't be able to hop onto Zoom, so it's going to have to be a remind message. And then if y'all have any questions, just let me know. Otherwise, I look forward to seeing what y'all come up with, and I will see y'all Thursday.